Hi, this is Ajit here. Today we are going to discuss phylum Ciliophora. Ciliophora comprises of a large group of highly diverse protists characterized by the presence of cilia, and cilia is used for movement and feeding. Ciliates may be marine, freshwater or soil dwelling, including extreme environmental conditions such as Antarctic ice and hot springs. All ciliates are heterotrophs and about 1/3 of them are ecto or endocommensals or parasites. The body of ciliates are covered by a very complex pellicle, and it consists of an outer membrane and an underlying single layer of membranous flattened sacs called alveoli. They usually possess a mouth or a cytostome and a cytopharynx. Ciliates have two kinds of nucleus, a macronucleus and a micronucleus. Therefore, they exist nuclear dimorphism. The macronucleus is typically larger than the micronucleus and contains thousands of copies of the genome. It is transcriptionally active and controls all functions of the cell and normal physiology. The micronucleus is smaller and is usually diploid, and it is considered as the germline reserve of the organism. It is similar to sperm or egg cell nuclei of multicellular organisms. The cilia may be arranged in rows, which may be longitudinal or spiral or may be in tufts. Cilia also have complex fibrillar system called the infraciliature. The infraciliature are usually found below the cell surface. The locomotor units or kinetids are composed of the cilia, a basal body and the associated fibers. Ciliates undertake asexual reproduction in a process called conjugation, which is typically a temporary fusion of the two partners through a conjugation canal. The two partners usually have to form complementary mating types. Once they have fused, they exchange the gametic nuclei. After this exchange, the two partners separate and the gametic nuclei will fuse and form the zygote, more specifically the zygotic nucleus. The zygotic nucleus then divides, the daughter nuclei produced develop into new macronucleus and a new micronucleus. About 8,000 species have been described by scientists so far. The most common example is Paramecium. There are other examples like Balantidium, Didinium and Vorticella. Now we will see the details of the Ciliate Paramecium. Paramecium is also known as slipper animalcule. Animalcule, as I had described earlier, are microscopic organisms. It's an old term used for microscopic organisms. This organism is called slipper animalcule because of its resemblance to the sole of a slipper. Paramecium occurs in freshwater systems like lakes, ponds, ditches and so on. It is a large protist measuring about 0.3 mm in length. A few Paramecium, if it is large enough, can be seen by naked eye. The body of Paramecium is flat with a blunt and rounded anterior end and a pointed posterior contractile vacuole end. It is called contractile vacuole end because at this end is where the contractile vacuole is present. The outer surface of the animal is lined by a pellicle, and the pellicle has rows of membranous sacs called alveoli. From each alveolus emerges a cilium. Each cilium arises from a basal body embedded in the cytoplasm below the pellicle. The cilia are arranged in longitudinal rows called kinetids. A kinetic is a row of repeating units called kinetids. Each kinetid consists of a cilium, a basal body and associated fibers called kinetodesma. All the kinetids in a ciliate are collectively called the infraciliature. The infrasiliary system controls and coordinates the movement of the cilia. The coordinated beating of the cilia results in a spiral forward movement of this organism. Extending obliquely backwards from the anterior end is a shallow groove called the peristomial groove or oral groove. It is lined with longer cilia, and the peristomial groove leads into a tubular cytostome or gullet which extends into the interior of the organism. The cytoplasm of the paramecium is differentiated into an outer clear ectoplasm and an inner granular endoplasm. The ectoplasm bears numerous bottle-shaped organelles called the trichosis. They are arranged perpendicular to the body surface. They alternate with the alveoli, and these trichosis are used for defense and for capturing the prey. When discharged, the trichosis rapidly ejects a very long thread-like shaft surrounded by a barb. During feeding, the food is driven into the peristomial groove and then into the cytostome by the cilia surrounding the oral groove. At the tip of the cytostome, a food vacuole is pinched off into the cytoplasm during feeding. The detached food vacuoles then begins a more circular movement through the endoplasm. During this period, digestion occurs. The food vacuole containing waste materials then moves to a fixed site called the cytoproct, where it fuses with the cell membrane and expels its contents. The residual vacuolar membrane then move to the cytostome, where they reconstitute a food vacuole again. Two contractile vacuoles are present in paramecia, which are the organelles of excretion or osmoregulation, and they are situated at the opposite ends of the body. Each vacuole consists of a central bladder and about 6 radiating canals. The contractile vacuoles are usually found in the ectoplasm, and these radiating canals of these contractile vacuoles will collect the excretory products from the surrounding cytoplasm and releases into the central bladder. The bladder consequently swells up and empties the contents to the outside through a permanent pore in the pellicle. Paramecium exhibits nuclear dimorphism, as I have discussed earlier. That is, there are two nucleus. One is a large macronucleus, which is in fact a kidney-shaped nucleus, and a small spherical micronucleus. The macronucleus controls the daily metabolic activities, and so it is also called the vegetative nucleus. The other nucleus is called the micronucleus and is concerned with sexual reproduction. Thank you for watching the video. Hit the like button if you love the video and click the bell icon for notifications.